Welcome, everyone, to the Deep Dive. And hey, a really special welcome today to Mr. Cheese students and all his TikTok followers and YouTube subscribers. It's awesome to have you here with us. Today, we are diving deep into something truly fundamental, the wonder of understanding life itself. Yeah, we're talking genetics and inheritance. Our mission really is to unpack the most important bits of knowledge from this fantastic guide that Mr. Chi shared, specifically for grade 12 CFPS students. We want to help spark those aha moments, make the complex stuff feel clearer for everyone, whether you're just starting out, maybe feeling a bit stuck, or if you're one of the high flyers looking for that extra insight, we're here for you. And yeah, this deep dive is possible thanks to those brilliant resources from Mr. Chi. So definitely remember to check out and follow his platforms for more great stuff, more study tips, really helps you get ready for exams. Okay, let's unpack this.
Yeah, that's a great way to start. Genetics, at its heart, it really is the study of how life passes down characteristics and all the variations that make everything so diverse. It's pretty revolutionary how far we've come, right from Mendel's early work all the way to gene editing today. It's just constant discovery. So yeah, our goal is to make this whole topic, which can seem intimidating, really accessible, bridge that gap, like you said.
Absolutely. So if we're talking fundamentals, the absolute basics, where do we start? How do you explain genetics at its simplest level?
Well, fundamentally, genetics is the science of heredity, right? How traits get passed down from parents to kids. And it's also about variation, why we're all different. It underpins basically all of biology, why you look like your parents, how species evolve. Yeah. It all comes back to genetics. And like I said, the field just keeps expanding. From Mendel sorting peas to sequencing entire genomes, it's been quite a journey, revealing life's blueprints.
Blueprints of life, like that. Yeah. So inside these blueprints, what are the core components we need to know? We hear about genes.
Exactly. Genes are the basic units. Think of them as individual instructions or recipes for specific traits, eye color, hair type, that sort of thing.
Okay, so recipes. But they aren't just loose pages floating around, right? They're organized somehow.
Oh, incredibly organized. These genes, these recipes, are located on structures called chromosomes. You can think of chromosomes as the cookbooks that hold all those recipes.
Cookbooks, got it.
And the language used in those cookbooks, the actual instructions themselves, that's DNA. Deoxyribonucleic acid. It's this amazing molecule that carries all the genetic instructions for, everything an organism does. Development, functioning, reproduction. What's really mind-blowing is that it's basically a universal language across almost all life on Earth.
Wow, a universal language. Okay, yeah. So understanding genes, chromosomes, DNA, how they all fit together, that's key.
Absolutely crucial. It's the foundation.
Now, you mentioned recipes or instructions. Do they always come in just one version? Or is there variation there too? That brings us to dominance and recessiveness, right?
Yeah, precisely. For a lot of traits, you get different versions of a gene. These are called alleles. Now, a dominant allele, it dominates. You only need one copy of that allele for the trait to show up. Like if dark hair is dominant, just one dark hair allele means you'll likely have dark hair.
Okay.
But a recessive allele is a bit more retiring. It only gets expressed if you have two copies of it. So using the hair example, you'd need two blonde hair alleles to actually have blonde hair.
Right. Okay. That makes sense. And that leads perfectly into something else that can sometimes trip students up. The difference between genotype and phenotype. Can you break that down?
Sure. Your genotype is your actual genetic code. It's the specific combination of alleles you inherited, like DAD or DT in our hair example. It's the underlying blueprint. Your phenotype, on the other hand, is what we actually see, the observable trait. So dark hair, blonde hair, that's the phenotype.
So genotype is the instruction, phenotype is the result.
Exactly. And if we connect this to the bigger picture, phenotype is influenced not just by the genotype, but often by the environment, too. Think about height genetics plays a huge role, but nutrition does too.
Good point. So it's nature and nurture influencing the phenotype.
Precisely. It explains so much of the diversity we see around us.
Fascinating. Okay, let's step back in time a bit because before we knew about DNA, someone was figuring out these patterns. Here's where it gets really interesting. Gregor Mendel, the father of modern genetics. What was so special about his work with pea plants?
Oh, Mendel's work is just foundational. He was a monk working in a monastery garden, and he was incredibly meticulous with his pea plants. He wasn't just observing some are tall, some are short. He was systematically crossing them, counting the offspring, tracking specific traits like flower color or pea shape over generations.
So it was the rigorous approach.
Absolutely. That careful observation and, importantly, his quantitative analysis, the counting, allowed him to figure out the basic rules, his laws of segregation and independent assortment. And this was way before anyone knew what a gene or DNA actually was. It's remarkable.
He used specific types of experiments, right? Monohybrid and dihybrid crosses?
Yes. Monohybrid crosses were where he focused on just one single trait at a time, like crossing a plant with purple flowers with one with white flowers and seeing what happened in the next generations.
Okay, one trait.
Then, dihybrid crosses involve looking at two traits simultaneously, so maybe flower color and pea texture, smooth or wrinkled. Tracking two things at once helped him work out more complex rules, like how different traits are inherited independently.
And to help visualize all this and predict the outcomes, there's a tool that students watching probably know. The Punnett square.
Yes, the Punnett square, a very useful tool. It's essentially a grid that helps you predict the potential genotypes and thus phenotypes of offspring from a particular cross. You basically map out the possible alleles from each parent and see all the potential combinations for their children. It makes calculating probabilities much easier.
Let's try a quick example, sort of based on the guide Mr. Cheese shared. Let's stick with hair color. Say, Dad has dark hair, but he's heterozygous, meaning he has one dominant D allele and one recessive D allele. His genotype is D-Day.
Okay, D father.
And Mom has blonde hair, which is recessive, so she must have two recessive alleles. Her genotype is DD.
Right, DD mother.
So setting up the Punnett square, Dad's D and D go across the top, Mom's D and D go down the side. In the first box, we combine Dad's D and Mom's D. We get DD, dark hair phenotype.
Yep.
Second box, Dad's D and Mom's D, we get DD, blonde hair phenotype.
Correct.
Then the next row just repeats that, right? DD again and DD again.
Exactly.
So what does this all mean for their kids?
Well, it means there's a 50% chance or probability of 2 out of 4 that a child will have the DD genotype and therefore dark hair.
And a 50% chance, 2 out of 4, of the DD genotype, meaning blonde hair. A 1-to-1 ratio in terms of probability.
Precisely. And understanding that probability is absolutely fundamental to genetics. It's how we interpret genetic data and make predictions.
It's amazing he worked that out just by counting peas. Okay, so Mendel gave us these foundational rules. But life isn't always that straightforward, is it? Sometimes inheritance patterns are more complex.
That's very true. We call this non-Mendelian inheritance. Mendel's rules are a fantastic starting point, but there are definitely exceptions and variations.
Like what, for example?
Well, one example is incomplete dominance. This is where the heterozygous phenotype is actually a blend, somewhere between the two homozygous phenotypes. Think of crossing a red snapdragon flower with a white one and getting pink flowers. Neither red nor white is fully dominant.
A blend. Okay.
Then there's codominance. Here, both alleles are fully and separately expressed in the heterozygote. The classic example is the human AB blood type. Someone with AB blood doesn't have blended A and B characteristics. They actually express both A and B antigens on their red blood cells.
So not blended, but both showing up fully. Got it.
Right. And things get even more layered. You can have multiple alleles where a gene has more than just 2 possible allele options in a population. Human ABO blood groups, again, you have the A allele, the B allele, and the O allele.
Okay, more than 2 choices.
And many traits aren't controlled by just one gene pair. That's polygenic inheritance. Think about human height or skin color or even intelligence. These are influenced by the combined effects of many different genes, often interacting with environmental factors too. That's why you see such continuous variation in traits like height.
Right, it's not just tall or short, there's a whole spectrum.
Exactly. And then there are sex-linked traits. These are traits determined by genes located on the sex chromosomes, usually the X chromosome in humans.
Why does that matter?
Well, because males have one X and one Y chromosome, while females have 2 X chromosomes. This means males are more likely to express recessive X-linked traits because they only have one copy of the X chromosome. If they inherit a recessive allele on that X, there's no second X chromosome with a potentially dominant allele to mask it.
Okay, like colorblindness, for example.
Colorblindness is a perfect example, yes. Hemophilia is another. And just to add one more layer of complexity, there's something called epistasis. This is where one gene can actually interfere with or mask the expression of a completely different gene. It's like a genetic override switch.
Wow. Okay, so it's definitely not always simple dominant recessive. There are blends, shared expression, multiple genes, sex differences, even genes overriding others.
It really highlights how complex and interconnected our genetic systems are, but understanding these variations helps explain the incredible diversity of life we see.
It really does. Okay, let's shift gears a bit. We've talked about the patterns, but let's go back to the physical stuff. That amazing molecule, DNA, and its structure, the double helix.
Ugh. Watson and Crick's discovery. Truly a pivotal moment in biology. Finding that elegant double helix structure instantly suggested how DNA could store vast amounts of information in the sequence of its bases, like letters in a code.
And crucially, how it could be copied, right?
Exactly. The two strands are complementary. If you know the sequence of one strand, you automatically know the sequence of the other. This complementarity is the key to DNA replication.
And how does that replication work? In simple terms.
Well, it's a complex biochemical process, but essentially the double helix unwinds, and each strand serves as a template for building a new complementary strand. Enzymes do the work of adding the right building blocks, nucleotides, and proofreading. The end result is two identical DNA molecules, ensuring that when a cell divides, each new cell gets a complete and accurate copy of the genetic instructions.
It sounds incredibly precise, but errors can happen.
They can, although this system is remarkably accurate. These errors, or changes in the DNA sequence, are called mutations.
And what are the consequences of mutations?
It really varies. Some mutations might have no effect at all. Maybe they occur in a non-critical part of the DNA, or they don't change the protein that's ultimately made. Others might cause minor changes. But some mutations can have significant effects, leading to genetic disorders or altering an organism's traits. But it's important to remember, mutations are also the raw material for evolution. They introduce genetic variation, which is essential for populations to adapt over time. So they're not always bad.
That's a really important perspective. Okay, so we have this DNA blueprint. It gets copied. How does the cell actually use the instructions in the DNA to build things, specifically proteins? This is protein synthesis, right?
Exactly. Protein synthesis is how the genetic information encoded in DNA gets translated into functional molecules, proteins, which do most of the work in our cells. It's a two-step process.
2 steps. What are they?
First is transcription. This happens inside the cell nucleus. A specific segment of DNA, a gene, is read and used as a template to create a complementary molecule of messenger RNA, or mRNA. Think of mRNA as a temporary, portable copy of that gene's instructions.
Okay, so DNA to mRNA is transcription.
Right. Then, that mRNA molecule travels out of the nucleus to the cytoplasm, where it encounters ribosomes, the cell's protein-making machinery. This is where step 2, translation, occurs.
Translation. Translating the message.
Precisely. The ribosome reads the mRNA sequence in groups of 3 bases called codons. Each codon specifies a particular amino acid. The ribosome links these amino acids together in the order dictated by the mRNA sequence, building a polypeptide chain, which then folds into a functional protein. And a key concept here is the universality of the genetic code. The same codons specify the same amino acids in nearly all organisms. It's powerful evidence for a common ancestor for all life.
Incredible. So transcription in the nucleus, translation of the ribosome, DNA to mRNA to protein. That's the central flow.
That's the core process, yes.
And just like errors can happen in replication, I assume mutations could affect protein synthesis too.
Yep.
So what does this all mean for the protein itself if there's a mutation in the gene?
Absolutely. A mutation in the DNA sequence can lead to a change in the mRNA sequence. During translation, this might result in the wrong amino acid being inserted into the protein chain.
And what does that do?
Well, depending on the change and where it occurs, it could make the protein less effective, completely non-functional, or sometimes even give it a new, potentially harmful function. In some cases, a mutation might create a stop signal too early, leading to a shortened, incomplete protein. It really highlights how crucial that genetic accuracy is for making functional proteins.
It really underscores the precision needed at the molecular level. Okay, this leads us naturally into a really modern area, how we've learned to not just understand, but actually manipulate genes. We're talking genetic engineering and biotechnology.
Yes, this field has exploded. It's about the direct manipulation of an organism's genes using biotechnology. We've developed techniques like recombinant DNA technology, basically cutting and pasting DNA segments, and gene cloning.
And there's one technique that gets talked about a lot now, CRISPR.
CRISPR-Cas9, yes. That's been a game changer. It's often described as like a molecular search and replace tool for DNA. It allows scientists to make very precise edits to the genome, cutting out faulty bits, inserting new sequences with unprecedented accuracy and relative ease compared to older methods.
Wow. And the applications of these technologies seem enormous.
They really are. In medicine, we're seeing the development of gene therapies aimed at correcting genetic disorders. We also use genetically modified organisms, like bacteria or yeast, to produce vital medicines like insulin or vaccines.
And in farming.
In agriculture, genetic modification has led to crops with enhanced traits, maybe resistance to pests or herbicides, improved nutritional value, or better yields. These are GMOs, genetically modified organisms.
Right.
And even in industry, genetically modified microbes are being engineered to produce biofuels or help clean up pollution. The potential applications are incredibly broad.
But with great power comes great responsibility, right? There must be ethical and social implications to consider here.
Absolutely. This raises some really important questions, and there's ongoing debate. Concerns about the long-term safety of consuming GMOs, although the scientific consensus is largely that currently approved GMOs are safe. There are also worries about potential ecological impacts, the possibility of genetic discrimination if genetic information becomes widely accessible, and perhaps most profoundly, the moral implications of editing the human germline, making changes that could be passed down through generations.
Yeah, those are huge questions.
They are, and they require careful consideration, public discussion, and thoughtful regulation. It's something future scientists, like many of Mr. Chi's students listening, and society as a whole need to grapple with.
Definitely food for thought. Now bring this back to humans. How do we study human genetics and inheritance patterns specifically? One tool you mentioned earlier was pedigree analysis.
Yes, pedigrees are very important in human genetics. They are essentially family trees that track the inheritance of specific traits or genetic disorders through generations.
How do they work? What do the symbols mean?
Standard symbols are used. Squares represent males, circles represent females. Lines connect parents and offspring. If an individual is affected by the trait being studied, their symbol is usually shaded in. Sometimes carriers of a recessive trait, individuals who have one copy of the allele but don't show the trait, are marked with a half-shaded symbol or a dot.
And by looking at the pattern across the generations.
You can often deduce the mode of inheritance. Is it dominant or recessive? Is it linked to the X chromosome? Pedigrees help answer these questions and are crucial for understanding how genetic conditions run in families.
And this helps us understand common genetic disorders like cystic fibrosis, sickle cell anemia, or Huntington's disease.
Exactly. We know the specific gene mutations that cause these conditions, and pedigree analysis helps illustrate their characteristic inheritance patterns, recessive for cystic fibrosis and sickle cell, dominant for Huntington's, for instance.
So for families potentially affected by these conditions, what resources are available?
Genetic counseling and testing are key. Genetic counselors are trained professionals who can help individuals and families understand their risk of inheriting or passing on a genetic condition. They explain complex genetic information, discuss testing options, interpret results, and provide emotional support. It's really about empowering people to make informed decisions based on their unique situation.
That sounds incredibly valuable.
Yeah.
Okay, we've covered theory, mechanisms, human applications. What about the practical side for students? Practical applications and experiments in genetics.
Yeah, getting hands-on is important. Students often learn fundamental laboratory techniques. Things like DNA extraction, getting the DNA out of cells, maybe from strawberries or cheek cells.
I remember doing that.
Yeah. Then there's PCR, the polymerase chain reaction. This technique lets you make millions or billions of copies of a specific DNA segment. It's like a genetic photocopier, essential for analyzing small amounts of DNA. And gel electrophoresis, which is a way to separate DNA fragments based on their size. You run an electric current through a gel matrix, and smaller fragments move faster and further. It lets you visualize DNA. And finally, DNA sequencing, which determines the exact order of the A's, T's, C's, and G's in a piece of DNA.
These techniques sound powerful, and they lead to real-world results, right?
Absolutely. We see case studies and real-world examples constantly. Like we mentioned, bacteria engineered to produce human insulin for diabetics. That was one of the first major successes of genetic engineering. Or developing crops resistant to certain pests, reducing the need for chemical pesticides.
So for students actually doing experiments, any quick tips?
Well, the basics are crucial. Follow proper laboratory protocols meticulously. Keep accurate records what you did, what you observed, safety first, always. And understand the principles of good experimental design, having control groups, repeating experiments for reliability, replication. These aren't just classroom rules. They're fundamental to good science.
Great advice. Wow, we've covered a lot. From the absolute basics, genes, chromosomes, DNA through Mendel's P's, the complexities beyond Mendel, the central dogma of DNA to protein, and right up to genetic engineering and its impact.
It's a huge field, and what's fascinating here is how rapidly it continues to evolve. Future directions in genetic research are incredibly exciting. We're moving towards truly personalized medicine, tailoring treatments based on your individual genetic profile. Gene editing technologies like CRISPR are becoming even more sophisticated, and we're still learning so much from the ongoing exploration of the human genome and the genomes of countless other species. The potential for discovery feels almost limitless.
And for the students listening, especially Mr. Chi's grade 12s, you guys are perfectly positioned. The knowledge you're gaining right now, it's the foundation you need to potentially contribute to these incredible future developments. You could be part of solving the next big genetic puzzle. So yeah, it's been a deep dive, but hopefully a really useful one. Genetics is just so dynamic, constantly changing with so many opportunities.
Definitely. So keep studying, stay curious, maybe explore some research that interests you, or even just keep up with the news on advancements. Your engagement truly is what will drive the future of this field.
And before we wrap up, a huge, massive thank you again to Mr. Chi for sharing these amazing resources that made this deep dive possible. And thank you to all of you, his fantastic students and followers on TikTok and YouTube. Seriously, remember to like, share, and hit that subscribe button on Mr. Chi's channels. Keep learning, keep exploring those study tips, and maybe leave this with a thought. As you continue your studies, what kind of genetic mystery might you be the one to unravel someday? Or maybe, what ethical challenge in this field will you help us navigate? Lots to think about. Thanks so much for joining us on The Deep Dive.